My name is Sasha Bronovitskiy. This past summer, I had the privilege of working in Dr. Fang-Ju Lin's lab as a research assistant through the CCU INBRE program. It's my honor to introduce the olfactory conditioning chamber design I've been working on this summer, with the help of Dr. Lin, Dakota Castillo, and Michael Yan. According to the NIH, Alzheimer's is a neurodegenerative disease characterized by neuron loss of function, brain atrophy, and memory impairment. Amyloid beta is a protein that could be cleaved to form the toxic fragment A-beta 42. A-beta 42 could then aggregate in the brain as Alzheimer's manifests. Studies, like the one from the ALZHEIME'S group, suggest a positive correlation between A-beta 42 presence and the severity of Alzheimer's. Here in Dr. Lin's lab, we study the toxicity of A-beta 42 using Drosophila melanogaster, or the household fruit fly, as a transgenic model. Drosophila is genetically similar to humans by a degree of about 60%, making them an ideal model to study human disease. Our control fly line, which contains the ELOV neuronal promoter and the GAL4 transcription factor, is crossbred with an upstream activating sequence A-beta 42 line, resulting in a fly that expresses the human A-beta 42 peptide and displays symptoms similar to humans' Alzheimer's. This fly line allows us to thoroughly study the physical and behavioral effects of Alzheimer's. If you take a closer look at the center image, you'll see a section from an Alzheimer's fly head displaying localizations of the A-beta 42 protein. This comes from another project from Dr. Lin's lab done by Michael Yan, where A-beta 42 aggregates were examined in vivo. Part of our research in Dr. Lin's lab is studying how Alzheimer's impacts memory as it progresses. It's known that Alzheimer's flies experience a deficit in short-term memory similar to humans with the condition. A popular research method used to quantify memory in Drosophila is the olfactory conditioning assay, in which flies are exposed to a shock and a unique scent simultaneously, followed by a different scent without a shock. This is described in papers from the Malik, Hayam, and Reisner groups. When presented to both scents after conditioning, control flies consistently select the scent presented without a shock, while Alzheimer's flies make variable scent selections as they aren't able to remember conditioning. We chose to use the compounds 3-octanol and 4-methylcyclohexanol for our scents, as the two are widely used in olfactory conditioning assays for their detectability but not overpowering attractiveness to flies. In our protocol, we note that these scents must be alternated when pairing with shock to ensure the flies have no intrinsic bias to either scent. We employed a 60-volt shock while conditioning, as this is enough to alert the flies without killing them. As depicted in this graphic from the Malik group, the T-maze is a device most traditionally used in research for olfactory conditioning assays. It's characterized by an elevator mechanism that transfers flies from the shock training point to the scent selection point. However, the T-maze tends to cause excessive fly casualties, as flies are crushed by the wall surrounding the elevator. In our previous study, Alyssa Reisner and her group successfully addressed the T-maze issue of fly casualty by introducing a two-part new generation olfactory conditioning chamber, in which flies are conditioned in a specific shock chamber, then transferred via tube and gentle airflow to the selection point where they make their scent choice. However, no airflow system was established to guarantee the scents reach the flies in each chamber. Additionally, we hypothesized their method of anesthetizing flies before inserting them into the chamber, coupled with their airflow transfer method, could potentially reduce fly alertness, as too much bodily stress causes flies to be sluggish and not particularly inclined to make a choice. For an overview of our materials, we used a 3D printer to fabricate our singular chamber design and slider. We utilized the Reisner Group scent tube design, characterized by a wire wrapped around cotton soaked in each scent. We added a pipette tip to a cut opening at the bottom of the scent tube to streamline air pump attachment, and a copper mesh insert to ease fly collection. For our preliminary trials, we used a copper-printed circuit board made by the Reisner Group. Feel free to pause the video for a more in-depth look into the materials used. Let's take a look at our conditioning setup. We used a 60-volt power source that delivered 12 shocks at 1.25 seconds each, with 3.75 seconds in between shocks. The shock sequence was determined by an Arduino Uno R3 microcontroller programmed by the Reisner Group. The cotton in each scent tube was soaked in 50 microliters of scent dilution. 3-octanol was diluted to a 25 millimolar concentration, and the 4-methylcyclohexanol to a 32 millimolar concentration, both resembling a 1 to 250 dilution in mineral oil. We plan to use an infrared light for our trials to eliminate any influential visual stimuli. We fitted our vacuum with a plastic nozzle so it could be placed snugly into the chamber opening. The air pump is connected to a controllable valve tube system, and each scent is assigned its own tube to prevent scent mixing. Every device is plugged into a power strip equipped with an on and off switch for optimal control. Now, to examine the first part of our single chamber design. Our main chamber contains a cubicle opening which the shock insert fits snugly into. The bottom insert, along with the printed circuit board, is first placed in the chamber to align exactly with the circular openings on each side, and is then followed by the top insert. Note the rectangular cutout on the side wall of the chamber that accommodates the wires of the printed circuit board. We cut holes in two 15 milliliter tube caps so that our scent tubes could be easily screwed onto the chamber. Next, onto our slider design. Our slider has 7 settings. First is setting 1, where live flies are directed into the chamber. Note the solid walls of each half setting on the slider. These are for rest periods during conditioning where we don't want flies to be exposed to scent or any other external stimuli. Setting 2, the training point, contains a singular copper mesh opening, so flies can be exposed to scent during conditioning without traveling into the scent tube. Both openings at setting 3 contain copper mesh inserts so that the vacuum can effectively remove each scent and introduce fresh air into the chamber. Setting 4, the selection point, has 2 unobstructed openings so flies can freely choose between scents after conditioning. Our olfactory conditioning protocol was heavily influenced by the Malik and Reisner groups, but we had to make some slight adjustments to accommodate our new chamber design. There are 6 main steps. First, 20 live flies were knocked into the chamber using a funnel. The flies had 90 seconds to accumulate inside the chamber. Then, flies were introduced to the first scent and shocked simultaneously for 60 seconds. That scent was promptly removed with a vacuum for 30 seconds. After, the flies were exposed to the second scent without a shock for 60 seconds. Again, the second scent was removed with a vacuum for 30 seconds. Finally, flies were given a 2-minute selection period. Each step corresponded to a specific setting on the slider. I know this is a bit much to process the first time around, so next we'll watch a video demonstration of the protocol. First, we begin with assembling our chamber. We insert the slider into the slots of the chamber, aligning the opening to setting 1 on the slider. Then, we connect the printed circuit board wiring to its power source. After, we tip the chamber on its side and place a foam pad underneath to ease fly insertion. A funnel is placed in the chamber opening. We locate and prepare our vial of imaginary flies. Then, the flies are knocked down into the chamber and the slider is promptly switched to setting 1.5 so the flies cannot escape. Flies are given a 90-second acclimation period while the power strip for all devices is switched on. Scent 1 is then screwed onto the chamber and a unique airflow tube is attached to the scent tube. The Arduino controller switch is turned on. The slider is moved to setting 2 and the power source is turned on. The flies are shocked for 60 seconds. The power source switch and Arduino controller are turned off and the airflow and scent tubing is removed. The slider is pushed to setting 3 and scent is removed with a vacuum for 30 seconds. Then, scent 2 and its unique airflow tube is attached to the chamber. They are exposed to scent 2 without a shock for 60 seconds. After, scent 2 is removed from the chamber with a vacuum for 30 seconds. The flies are then given a 90-second rest period on slider setting 3.5. After, both scent tubes are screwed onto the chamber. Note that we're not attaching airflow tubes to each scent this time because we don't want scents to mix inside the chamber. The flies are left to choose their scent for 2 minutes on slider setting 4. Lastly, flies are collected by anesthetizing each tube with CO2. And now, an imaginary drumroll please for our conclusion. Through 7 preliminary trials, our conditioning chamber setup killed only 2 flies out of the 80 plus used, so I can confidently say our design minimizes fly casualty. Our valve-controlled air pump system guarantees efficient scent dispersal, so we can absolutely be sure our flies are able to detect each scent during conditioning. Finally, our protocol minimizes fly trauma and therefore allows flies to be as alert as possible during conditioning. Now for future iterations. During preliminary trials, we decided to use the Reisner Group's printed circuit board. However, the circuit was fragile due to its age and broke through one of our trials. Because of this, we weren't able to collect acceptable data. Recently, we obtained the proper materials and protocol to make a reliable printed circuit board from scratch, which we plan to do in the coming weeks. These pictures depict our first attempt to etch a copper printed circuit board, using a design printed on toner paper and homemade ferric chloride solution. I really wish I could say this attempt was successful, but we're persevering. Once we make a reliable printed circuit board, we plan on using ALZHEIME'S flies in our conditioning assay. I'd definitely be remiss if I didn't thank Dr. Lin for all of her indispensable guidance throughout this very new experience for me. I'd also like to thank my co-authors, Dakota Castillo and Michael Yan, for their incredible contributions to this project. Furthermore, I must acknowledge the Reisner Group for their research and its fundamental importance in this project. Also, a thank you is owed to Dr. Wakefield for providing the 3-octanol used in our assay. Finally, the biggest of gratitude to the INBRE program and Coastal Carolina University, along with the organizations who funded us, for providing the resources to make this all possible. Thank you again so much for watching my presentation. Feel free to pause for citation and reference information in later slides.